So at the end of this week, we carried out an investigation with some pondweed where we were looking at the number of bubbles produced per minute when we changed the light intensity by moving a light further and closer to the pondweed. We ended up, all of us, drawing a graph that looked a little bit like this. We're going to pick up from this point. We've seen the graph showing how the light intensity affects the rate of photosynthesis, but we need to know how we would actually get the data for the graph. To investigate the effect of different light intensities on the rate of photosynthesis, you need to be able to measure the rate. One way to do this involves an aquatic plant, such as Canadian pondweed. The rate at which the pondweed produces oxygen corresponds to the rate at which it's photosynthesizing. The faster the rate of oxygen production, the faster the rate of photosynthesis. Here's how the experiment works. A source of white light is placed at a specific distance from the pondweed. You should then leave the pondweed for a couple of minutes to adjust to the new light intensity before starting the experiment. When you're ready to start, the pondweed is left to photosynthesize for a set amount of time. As it photosynthesizes, the oxygen released will be collected. Make sure you do a risk assessment before you start this investigation, so you carry it out safely. There's a slight risk of infection from the pond water, so make sure you wash your hands after handling the pondweed and cover up any cuts. Canadian pondweed is not native to the UK, so it must be disposed of carefully after the experiment. It shouldn't be thrown out and released into the environment. The experiment is repeated twice with the light source at the same distance, and the mean volume of oxygen produced is calculated. Then the whole experiment is repeated with the light source at different distances from the pondweed. Taking repeat readings and calculating a mean reduces the effect of random errors and will make your results more precise. In this experiment, you investigated the effect of different light intensities on the rate of photosynthesis. The light intensity is the independent variable. The independent variable is the variable that you change in the experiment. The dependent variable, the variable you're measuring, is the amount of oxygen produced, which is represented either by the number of bubbles. You could measure the volume of gas produced. You could use a syringe and measure the length of the bubble that is produced. You have to try and keep all the other variables constant so that it's a fair test. The variables in this experiment include all of the limiting factors of photosynthesis. Ways you can control the variables. If your plant's in a flask, you could keep the flask in a water bath to try and keep the temperature constant. There's not much you can do to keep the carbon dioxide levels constant. You may just have to use a large container for your plant. Do the experiment as quickly as you can so the plant doesn't use up the carbon dioxide. Another way you could possibly control temperature is by placing a clear test tube or a clear tank of water between the pondweed and the lamp. Then this water will absorb the heat and still let the light through. You can compare the results at different light intensities by giving the rate of photosynthesis. Calculate the rate, just divide the total volume of the bubbles or the gas by the time taken to produce it. Number of bubbles per minute is what we did, but you could work it out by doing the volume divided by the time. If you measured the size of the bubble, you could do the length of the bubble divided by the time. When you're working out the rate, you should always be dividing by time. See if you can use all this information to answer the question that has been attached with this video on Google Classroom.